All right, everybody. So here is chapter 3. Oh, there's a go. All righty. Here I am, ready for you guys. I might move this around a little bit depending upon the slides. Didn't show up first. All right. So heading on to chapter 3. This is a pretty critical chapter, and I've actually added a couple of more details from slides that I've used in prior lectures with a different textbook for just providing a little bit more explanation and more figure options and things like that. So this chapter will be broken up into sections that correspond with the sections of the chapter. And the videos will be broken down according to those. So at the end of each section, I'll get to a questions screen, and that is where typically I would take questions, and we also stop the recording at that time and then start again, moving on to the next section. Alrighty, so we begin this section. We're going to really want to learn about neuronal communication. This is essential. You guys learned a little bit more about some of the basics of neurons in terms of their composition and things like that, and why they're important in biopsychology behavior. I mean, they are essential. It is what, like, all our cognitive thought and everything is built upon is neurons. So this, like at a bare sense, this next chapter, these two, in terms of understanding, neurotransmitters, our reliance on them and how they affect our behaviors is going to be really illustrative for understanding like a host of things in regards to medications. Medications will actually contain elements to activate the neurotransmitters, affecting sort of serotonin neurotransmitters and different types. So, and this is the way a lot of medications will activate through neurotransmitters, and that they can then affect hormones and cascading effects, and then also just trying to alter in subtle ways the way, medications will try to alter in subtle ways the way that neurons communicate to hide it, how to try to help balance sort of to help balance neurotransmitter systems like the dopaminergic system or the serotonergic system to keep in the right balance. Because when they are not in the right balance, and that they have a chemical imbalance, that's when you're going to experience some of the array of sort of mental health challenges. So we're going to begin by thinking of the principles of neuronal communication. And so the basic element underlying neurotransmission is electricity. So electricity is the flow of electrons from a body that contains a higher charge, more electrons, to a body contains lower charge, fewer electrons. And we'll give a little more example on the next couple of slides. So this electron flow from higher sort of charge to lower charge, this can perform work. As an example of your textbook, sort of like a light bulb. And these electrical impulses, when they occur if within biological tissue that contains an electrical charge, the charge can be recorded in a sense of how it's sort of experienced with the cell. And that charge can have sort of cascading effects. So if living tissue is sensitive to an electrical charge, then the tissue can be stimulated. And so this can be anything from sort of like a slight reflex action to things that are done as types of like specific mental health treatments and stuff like that to target sort of like electrical impulses to sort of specific sort of regions deep in the brain to kind of help, to help address like untreatable and unretractable depression. So, so there's so much of this that comes into things that you'll actually really experience. So we have this, we know that living electricity can travel over living tissue because people get struck by lightning and all that kind of stuff, and we can get electrocuted. And so, but here's some of the basics of how it looks across this nerve here. So for electrical stimulation, current leaves this sort of simulator, and this is basically the way they demonstrated that you could have this electrical charge occur across this nerve. So we know this now, but this is one of the original ways. So you think of this basically as aspects of a battery, and it's like attached right here, and then you put the electricity right here, and you see basically if it gets sort of recorded and it comes back, and it basically tells you, the intensity. Gives these stimulating electrode delivers sort of these millivolts, and these intensities are sufficient to basically produce a response without damaging the cell. So too high would damage it, but so they basically get this right proportion that generally aligns with, the natural sort of the natural bandwidth that would work for sort of for this electricity sort of passing down nerves. And so then you can basically record this. So they were able to do this experiment and show like, okay, electricity actually flows down here. And so then they started to say, okay, well, if we're going to take this to the next level, what does some of this mean? So here, if we have this electricity sort of that charges down, well, how, if this impulse travels down, it's going to communicate information. How does that, that travel across the sort of the neuron? How does it work? What happens there? So taking a step back, you may remember some of this stuff from high school physics. We have this basic element with ions. Thinking way back, ions are charged particles, and they can either be positively charged cations, or which be sodium and potassium. These are things that are involved um, in neurotransmission and neurocommunication. And then we have anions, which are negatively charged ions, and we have chloride and then protein molecules that are also negatively charged. And so these are the ions that are involved in the key parts of sort of this neuronal communication process. So one of the most important things is to look at, okay, we understand these charged particles, and so what is happening within the neuron? And what we find when we first started to sort of look and to see how these things passage, that the cell membrane, can basically keep sort of one electrical, like basically differences in charge across the cell membrane. And so at one point here, then the inside can, increase to more positive, and then another point sort of it can decrease to more negative. The basic balance between the charge from the outside and the inside is negative 70 millivolts. So whatever reason they set 0 on the outside, and then how much more negative the inside is negative 70 millivolts. And so the cell membrane is very basically sensitive to changes in voltage across the membrane. And so this baseline here, this negative 70 millivolts, is known as the resting potential of the cell. So, and this is important. This is where it kind of returns to when it's at rest, the sort of negative sort of standpoint. And one of the ways might be able to think about it is that it's negative. So it's like, what does that mean? It could be like you're kind of pulling, I wish I had a rubber band, but if you ever sort of pull the rubber band back, and that keeps it taut, and it's right there. And it's like, oh, we got that right there. And it's like, ready for release. So that's kind of what you would want to think of this negative 70 millivolts sort of returns to that space. And it stays there ready basically for the sort of sudden release into positive that can cause downstream changes. Alrighty. So how do we get some of this differences between the inside and the outside when you have a membrane, a semi-permeable membrane, the cell membrane, that things can pass through? So there's two principles that underlie the difference in voltage between the inside of the cell, what makes it more negative, and the outside of the cell, which is less negative. It's not more, it's not, I mean, it's more positive, but it's still sort of set to zero, so it's just not as negative. So one, we have these two principles that are basically a concentration gradient and a voltage gradient, okay? So this concentration gradient, this is just like if you were to put ink or paint into, into a cup, and it would diffuse over time until it was basically equally distributed throughout all the water. So that's just what a concentrate does. It's going to automatically diffuse, and over time it'll all have to be equally presented. It's not like it's going to like more of us are going to hang out in this one corner over here. So that's one principle. And then you also have the voltage gradient, and we don't just have sort of water in there, this intracellular fluid. It has these differentially charged ions, and the and if we pour salt in, and looking at how that distributes, a salty solution has both positive and negatively charged ions. And what they will do is they will basically distribute sort of themselves and engage in sort of elements of diffusion as well. And then they will distribute themselves, because it has both negatively and positively charges over time, in this sort of makeup right here, which is like, okay, here, they bind together, positively and negative, ions, they kind of stick together. And so then they'll organize themselves basically in this electrostatic gradient of positive and negative charges, where everything kind of balances out, and it tries to balance it out to be the same relative voltage across all of within all of the liquid. Right, so now what happens, actually I'll go back. So then what happens is that if you have a semi-permeable membrane, which we're going to be learning about on the next slide, then what, like the concentration gradient tries to occur in terms of just spreading everything out equally, and the voltage gradient tries to occur. But the membrane will only let certain things sort of pass through. And based on sort of the size of it, then certain positively charged ions are too big, they'll have to stay on the outside, and there's no, and so then the small anions go sort of more on the inside, and they will negatively charge the cell, or you'll have chloride that sort of, that pushes these negative charges of the cell. And but then all of those pieces, the molecules and everything, have to follow this concentration gradient and voltage gradient. And so when you put up a semi-permeable membrane, you might have this inside of the cell that's negative is going to try to attract in positive things from the outside of the cell. And so that's going to kind of pull things in this way. But then the concentration gradient is still going to want to be like equal across, and they're still going to try to keep the sort of same voltage. So it basically can result in this distribution of electrical charge, where naturally, after the concentration gradient and the voltage gradient is sort of applied, then through this combination, there will be differences between the inside of the cell and the outside of the cell. So you can imagine if there were like a semi-permeable membrane right here that only let certain ones through, say more of the positively charged like potassium, then that would travel through, but then more negative ones would be over here on this side. But then you have more positive over here. Well, then they'll want to go ahead and come back together, like right at that membrane. And then they'll still be trying to, well, we want to, make the insides become more negative. We want to make it, more positive. So then the potassium might want to flow back inside, but then it messes up the concentration gradient. So these two pieces like work together to create sort of the flow of electrical charge between the intracellular and extracellular and this semi-permeable membrane. All right, so here is this, the next steps on this piece. Here is this, where we have our membrane. And as I've sort of talked about here, we have the extracellular fluid and the intracellular fluid. The extracellular fluid is more positive, and compared with the inside of the cell. And again, this resting potential, you have this sort of tautness of the rubber band, is at this sort of set to be at this typically right around negative 70 millivolts. Okay, so here is, and how you'll start to see how some of this voltage gradient and concentration gradients are applied. Alrighty, back up here. So the major players, sodium, there's more on the outside of the cell, potassium, more on the inside, chloride, which is more on the outside, and then we have large negatively charged protein molecules that are on the inside, which is these guys right here, the proteins that always just sort of stay mostly in the cell. So we have these ion channels right here, and the large proteins, these are large proteins in the membrane that selectively permit the flow of specific ions. So, and these specific ions, with the channel along sort of the whole axon, excuse me, then like this is the cellular sort of membrane, and these ions will let some stuff through in all of these particular portions of the cell, so that depending upon how they are stimulated, and then try to adjust the voltage level of the inside versus the outside of the cell. So these are voltage gated ions that will basically be activated as a certain portion of the membrane as around it becomes more negative or more positive. So these are charged, basically the voltage affects them, whether they open or whether they close. And then you have other ion channels, which are chemically gated. And this is an ion channel that opens up and closes by a chemical such as a neurotransmitter. And so then, and we'll be learning about these chemically gated channels in a second. And then these can be opened by like specific chemicals that fit into specific receptors. And so we'll be learning a little bit more about chemically gated as well. All right, so in the so what is happening then inside the cell when you're going to have this negative 70 voltage right here, and then how is that stuff going to change? So we're learning about these molecules that selectively let sodium and potassium through. And so the flow of these ions is going to give rise to depolarization of the cell, which is basically where the neuron membrane becomes less polarized, so less difference between the inside and the outside. And this is caused by the sodium ions entering into the neuron. And the voltage-gated sodium ion channels, they quickly open when that comes nearby. So you have a flood of the sodium in. And so when this sodium floods from the extracellular into the inside, then it changes the potential. The inside of the cell becomes more positive, as you can see right here. So it's sitting at negative 70. And then when the sodium starts to rush in there, then it flips up. It's like, oh, we're closer to negative 65, and then over time, they'll kind of return back to normal to go back down. Okay. Now, for hyperpolarization, that's the opposite, where it's going to actually become more negative. The neuron membrane becomes polarized again, caused by potassium exiting the neuron through the potassium channels that open more slowly. So then the potassium is differential between the inside and the outside of the cell. And so when those leave the cell, then it becomes even more negative, which makes it, which makes the neuron even basically less likely to fire. Because through depolarization, then you're going to, in a sufficient depolarization to sort of summate at the action potential, at the axon hillock, for example, then that will cause the action potential. Whereas hyperpolarization makes action potential less likely, depolarization makes action potentials more likely. Right. All right, so the time sequence of an action potential showing when ion channels open and close. So, what is an action potential? As we've sort of been leading up to having these voltage differences, an action potential is a temporary reversal of membrane polarity caused by voltage-gated ion channels reaching their threshold, okay? And so what happens here is this is the threshold. So when you get down to this, you're at this -70, but when you have this sufficient depolarization in the space that's closest to the axon hillock, or it gets to that space, then it's, if you can get it to sufficiently depolarize to -55, then that is what the threshold is for basically beginning action potential. Because at the -55 threshold, so you don't even have to go down to 0, you just have to go become really sufficiently less negative. Then at that point, when the inside of the cell reaches that level, then these sodium ion channels, they basically sense the voltage, they open, and then the sodium basically floods into the cells we talked about, and you have this rapid depolarization that is basically the action potential. It looks here and it follows this form. And so along this process here, you have the sodium channels open, then the depolarization occurs. Once you get sort of sufficiently, then you have up here, you're going to have different sort of voltage channels. At this point, the potassium channels open, and then potassium, starts to exit the cell, and then the sodium channels close, and so no more sodium is sort of reaching the cell. So this is the point where it generally becomes the most depolarized, and then the effects of these closing and opening, then basically those that process, the opening at this particular stage and the sodium flowing in, and then the potassium flowing out, and then the sodium channels closing, then this causes the repolarization. And at this time, the potassium channels, they're open, and the potassium is continuing to sort of exit and making the membrane, the membrane potential more and more and more negative. And then at this point, -70 is when the potassium channels close. And so then that is to return it back to this resting state. Then sometimes, though, during that process, even though they close, it can be more negative, and then take time for this, you have this sort of period past here where it's after the action potential, but it's still sort of trying to return to resting state. And so the other piece that you can see here, we actually switched. And so what happens here, I was like, wait. So we've talked about this action potential going down and through this process, which is uses the same exact figure. So sorry about that. Then this threshold, this -55, that has to happen at a particular location, which is called the axon hillock. And this is where the axon meets the cell body that contains the high density of ion channels. So the cell body, if we would go back to our little diagram, we have the cell body here, and then usually the axon sort of sticks out sort of the bottom here, goes these various places, and right where the cell body and the axon meet, that's the axon hillock. And all of the for an action potential to occur, depolarization has to reach that axon hillock, regardless of which place that it starts on the cell body. It will need to reach that place in the axon hillock that contains the axon initial segment. And this is the segment of the axon just past the axon hillock, where an electrical signal of the actual action potential begins. So it's like you hit the axon hillock, and then we've registered stuff, and then the actual action potential is basically on the other side of the axon hillock, cell body on one side, the axon initial segment there, and then that's sort of the technical begin of the action potential traveling down the axon. And so, as we talked about at this point, this axon initial segment, you can have depolarization, and this is a reduction in the membrane potential of a neuron caused by the channels opening and entering the neuron. That if this occurs at the initial axon segment, then you will have the action potential. And but then repolarization after this depolarization process is where everything returns back to negative. And then again, it can get hyperpolarized, right? Sort of down here with, potentially even having more sort of almost inhibitory, inhibitory cells could potentially, or inhibitory neurotransmitters could actually try to hyperpolarize a cell to make it even more negative, and to then have, neurotransmitters that have depolarizing effects that can actually make it a little bit harder. So a neuron can have tons of connections with other neurons that are excitatory or inhibitory, or could be both, depending upon what neurotransmitter is being released. Alrighty, so now where we're getting to this next one. So we've gone through the action potential, and sorry for that, the slide switch that I missed earlier, but you can see here that we have these sort of two phases. And so this absolute refractory period is basically the 1 to 2 milliseconds during an action potential when another action potential cannot be produced. So it's like, oops, sorry, we're in this one. It's like the toilet is flushing. You can't really make another flush. This one car roller coaster has started and you cannot start another one, right here, whatever kind of. Now, there's a number of helpful analogies to use, but basically within this time frame, the action potential in the cell is being used. It's sort of occupied, and it cannot become occupied by something else. But once the repolarization occurs down to the resting potential, so it's met this, and technically it's gone negative. Well, it can be depolarized and, come up here. It's harder to do so because it becomes hyperpolarized. But this is what's called the relative refractory period. It's a short time when the neuron membrane is hyperpolarized. It's more negative, but, and it's difficult, but it's not impossible to generate another action potential. But it will just need an especially powerful stimulus, or especially powerful stimulus at a particularly key locations. Alrighty. So now, once we've got this, we've talked about, okay, here's the setup. Okay, the action potential is occurring. Well, then what happens then? How does this action potential occur? So first, we have this sort of all or none law that largely aligns with this, strict refractory period. Once an action potential begins, it travels all the way down the axon, and it begins, that's kind of why another one can't get started, and sort of like you've got one electrical wave, and oh, we're going to have a heavier one over here, and it's just going to overtake it. That can't occur. It's just, down the axon, down the axon. And so it's all or none. Once the action potential starts, it travels all the way down. Then how does this travel all the way down? And it's going to use some of the same sort of principles of, this is basically like an action potential that has to continually go down the cell go down aspects of this terminal for the action potential to continue to travel down the axon to meet the other cell and have the effects that it wants to. So that can be a long way. Axons can be quite long, especially for when they're traveling from, within the central nervous system, where they can go all the way down the spine, or in the peripheral nervous system where they can go all the way down limbs. So what the process that kind of propagates these down the axon is called saltatory conduction. And this is what happens on myelinated axons. So myelination is extremely important for healthy brain and body functioning. So these the myelinated axons have these sort of periods of myelination right here. And then in between, they have these sort of nodes that are called nodes of Ranvier. And then what these do is they're kind of like the saltatory conduction are sort of points of like rapid flow. So it's like you hit here, have this activate here, you have the action potential occur, the cell body is back this way, this is the terminal that's sort of heading back down, you see the myelin right here, there's the nodes of Ranvier. So once in this saltatory conduction, what happens is that you basically have the action potential here, and this sort of voltage change in the sodium ions sort of coming in, then you basically can have this sort of jump, so instead of traveling all the way around, the myelin kind of lets the sort of like voltage go, sorry for the, or I can go, but it basically sort of hops down and can pass through these like highly conductive, the myelin is sort of highly conductive, and so it can jump down. That means the action potential can travel down the axon considerably faster. And so we really rely on that, particularly for motor functions and anything, because if we had to kind of rely on just the propagation of the action potential down the axon, continually the action potential propagating down, that's slower, and we wouldn't be able to move quickly like we would need to assist with our survival and dodging objects, playing sports, and a number of other things. So this is how they sort of jump down through this process of saltatory conduction. And then it can, and as these things, just as you have like any electrical sort of wire that is what does it mean, that is covered in sort of the, sorry, I'm blanking on the term, but it's covered on an insulation. And what that special insulation does is it actually helps the electricity sort of travel down with like zero resistance and sort of unimpeded. And that's kind of what the myelin is able to do too. It kind of increase the speed of the signal. So in kind of thinking, okay, we've got this action potential, it's occurred, but then what happens afterwards? How do we actually return back to some of this resting potential pieces? So after an action potential, the sodium and the potassium must be pumped against the gradient because of the voltage gradient and the concentration gradient. They must be pumped against the gradient back to their original location to return the inside of the cell to its negativity. So that's then the sodium-potassium pump steps in. What it does is it uses energy in the form of ATP. This is what this is like basically pretty much the energy that our body uses. This is what it's sort of called, and it's sort of more sort of molecular form. And so this energy, from eating, like from, being well rested and all those types of things, eating well, eating sort of, and living sort of healthfully in general, then you would be trying to, even when this is where the elements of food is actually sort of fuel for your body. If you are actually consuming sort of the food in terms of not just sort of its taste, but also its other sorts of properties, these are the nutrients that your body is taking in terms of using the molecules from your food to sustain yourself, to give yourself nutrients and to give yourself energy in this form of ATP. So what the ATP does is it's used by the sodium-potassium pump as energy to change the cell back to normal. So what happens then, here you have the extracellular fluid and the cytoplasm inside. And so sodium is low and potassium is high. So what it'll do here is the sodium will kind of climb back into these spaces right here. And the ATP comes and it attaches to this little molecule, turns into the ADP, both of those. And this then causes the protein to shift in shape, and just like this here. And then it releases the sodium ions back outside. And then when it's trying to basically say, okay, now we're returning to normal, it doesn't just take those back out, but it basically is like, look, those all attached to this side of this protein. And it has these other little spaces right here in the protein that these are actually ready for potassium molecules. So when the sodium leaves, the potassium comes in and attaches to these here. And then you have the result of this sort of ATP process. This sort of protein end up sort of collecting here and leaving. And when that does, it basically has it open this way, and the potassium leaves. It leaves the cell, and then it returns. Then it's basically in the shape that it is here again to bring out more sodium and to, return potassium. So this is what returns the cell to resting potential, to get the rest to basically continue neuronal communication. And if you don't have enough ATP and things like that, that is the stuff that'll eventually cause your cells to die. And they're not having some of the the energy, some of the pieces that they need. They could be starving. This is the stuff that can happen with imbalances of insulin, and like in type 1 diabetes, that insulin can get so out of whack that it doesn't realize you don't have any basically energy or sort of sugar and elements in your molecule in your bloodstream that cannot actually give your cells the nutrients and energy it needs to perform some of these needed functions. All right. So this is questions. That means this is sort of the end of that neuronal communication section. And now we'll be heading on to the next one, which is the synapse. So we'll be coming back to learn more about the synapse, which is again where, here you've got this action potential coming down, and like, well, this is the presynaptic terminal where it's gonna, this is what's gonna meet up with another sort of dendrite, and you can see some of these pieces here, and that's what we're going to talk about next. So see you guys next time, next video in a couple minutes.